Okay, good evening, everyone. Welcome to Storm Surge, and glad you're here on this little chilly night with us. I'm going to hope this works. There we go. So for those of you who are new to Storm Surge, first of all, I'd like to introduce myself. I'm Kathleen Britton. I'm on the board. Storm Surge, the speaker series has been one of our notable activities. Since 2013, we've, as you can read, have been addressing coastal issues of climate change. And we do this by promoting science-based education in a number of different ways, including our speaker series. We also have educational videos and other community event opportunities that are on the website. So, I'm going to hope this works. Ta-da! We would like to extend our gratitude to the Institution for Savings. It's sponsoring this year's speaker series. We also are acknowledging the city of Newburyport for the use of this very lovely facility here, and to the Newburyport Media Hub. They are filming this. It's live right now, and then we also post it on our website in a couple of days. So Storm Surge exists because of people like you who share our mission to empower our community with knowledge needed to actively mitigate and adapt to our rapidly changing shorelines, our coastal and marsh environments, and our local weather patterns. We are a volunteer-driven organization. We have no paid staff. Each year, the value of our volunteer hours equals about $10,000. So that means if you are financially supporting us through your donations, you can be assured that 100% of your donations go directly to our program. And so we thank you for your donations and for your participation and activism. Why is this not? We have a different computer this time. Wanted to alert you to mark your calendar for our speaker series that extends in the spring. On February 5th, we have Erica Spenger, the Union of Concerned Scientists. On March 5th, we have Emma Gildesgame of the Nature Conservancy. And on April 27th, we have a film, a documentary that we're presenting at thank you, I'm going to say it, at the screening room here in Newburyport. That will also feature a live Q&A with the filmmaker. So be sure to put these on your calendar, and we'll have more information on our website. So this evening, it's my pleasure to welcome Kurt Rogers, who is Executive Director of the MRWC, and Jose Tapia, who is their Water Quality Project Manager. Kurt brings over 20 years of nonprofit executive director experience to the MRWC. He has in-depth experience in strategic leadership, financial and program management, community engagement, and policy advocacy. He holds a Master of Public Administration degree from Harvard's Kennedy School of Government. And in his spare time, he volunteers with the Humane Society of the United States, both as an animal rescue volunteer and as a policy advocate. Jose Tapia first joined the MRWA in 2018, first as an intern and then as an environmental fellow to assist with the various digital education projects, as well as regularly collecting water quality data. He is now a permanent staff member working with the Water Rescues Program, and his goal is to engage communities in science, restoration, and environmental education. Jose holds a Bachelor of Arts degree in Environmental and Legal Studies from UMass Lowell. So it is my pleasure to welcome them this evening, and thank you for being here.
how does it feel to have your resume read out in front of you? It's a little formal, but thank you very much, and thanks for having us. Before I even get started, I just have to ask Jose, where is that picture taken? That's gorgeous.
That is Mount Tom, Massachusetts.
Mount Tom, Massachusetts. Okay, bear with me, folks. I have the honor here of hitting escape and switching over to a different slide deck. There we go. And working my way down here. Okay, almost. And one more. Full screen icon. Is it down here, I believe?
I think it's this guy. Nope, up here.
Up there?
Play button.
That guy?
Yes. Hey, there it is.
There we go. All right, thanks for bearing with me. So my charge tonight is to really tee things up for Jose. So tonight I'm going to share, I'm going to share some information about the Merrimack River Watershed Council. That's not good. I'm going to share some information about the Watershed Council. Oh. It's on a, yeah. That's not going to work, folks. Hang on. Let's exit the slideshow. There should be a full screen.
On the bottom right, there's a little arrow.
Yeah, I think that's, nope.
Oh, where'd you go?
I clicked right there, and that's what got us into that. If you probably could click, that's what I clicked. Put that up.
This guy right there.
Oh, I clicked below it. Thank you very much. Okay, now we're off to the races. Again, I'm going to give you a quick introduction on the Merrimack River Watershed Council. Then I'm also going to talk with a brief overview of what is a combined sewer overflow. Then we're going to get to the meat of why I think we're here, which is really to talk about our 2024 water quality data and what that's told us, especially about what's happening in Newburyport Harbor. That's what Jose is going to share with you, and then we'll open it up for some questions. I'm going to go kind of fast because I think what we really want to get into is Jose's material. If you have questions, I'm going to ask that we hold them to the end, because if you have questions about what I say, by the time you hear what Jose says, you're really going to want to spend your time asking him questions and not me. So we'll try to get through this fast. So we're going to start out with the core of what MRWC's focus areas are. They are restoration, water quality, regional prioritization, and policy advocacy. In our restoration work, part of that work is assessing the literally thousands of culverts that there are throughout the watershed. Culverts are stream crossings, and they can be problematic due to being undersized, inappropriately positioned, inadequately, and I'm going to have a correction. 1 second.
You lost your screen.
So culverts can be problematic due to being undersized, inappropriately positioned, or collapsed and failing. And these failures can reduce water quality, increase flood risk, and prevent fish passage. Standardized culvert assessment allows us to work with municipal partners to prioritize culverts for replacement and repair. Now, another part of our restoration work is supporting dam removal projects. There are over 1,500 dams in the watershed, most no longer serving any function, and many poorly maintained and at risk of failure. Like culverts, they can segment wildlife habitat and prevent fish migration. Now, the Merrimack's host to several diadromous fish species. Diadromous fish are fish that spend part of their lives in fresh water in the rivers and part of their lives out in the ocean. Two quick examples are river herring, which spawn in the spring in the fresh water of the river, then as adolescents swim out into the ocean, spend most of their lives, and then come back and spawn. American eels, on the other hand, do that in the reverse. And believe it or not, the Merrimack River used to host an active salmon run. So that's our restoration work in a nutshell. Our water quality work is focused on testing, and it's heavily reliant on our volunteer crew. We've 4 testing programs, and we also support a UMass Lowell research project on tap water testing. So one of our programs is our baseline monitoring program. The baseline program collects data about basic river health metrics. Think of it like going to the doctor's office and you always get documented your baseline temperature, your blood pressure, your height, your weight. So your pulse, it all gets documented. Well, we've been collecting our baseline data for years, and it's an important legacy data set. We currently test at 15 sites from Manchester to the mouth of the river. We test once a month for 4 months in the winter, and then in the other 8 months, we're testing twice a month. Our baseline monitoring program tests for pH, salinity, dissolved solids, temperature, conductivity, and fecal bacteria. The next of our water quality programs is our CSO monitoring program. We monitor for 3 days after a significant CSO. We use the same 15 sites. However, we only gather data on fecal bacteria levels. Due to the difficulty in responding on call to CSO activity and the significant number of people hours involved in a 3-day CSO test, MRWC only has had the resources to test 1 CSO per each 7-day period. Our next testing program is a pilot program to explore the new technology of cloud-based remote sensors. With a generous loan from the Lowell Duck Island Wastewater Treatment Plant, MRWC ran a sensor at the Newburyport Harbor Master Pier. We ran the sensor weekly to give results on Fridays to alert folks as to the water quality before the weekend. We also ran it before major holidays and after CSOs. So these first 3 water quality programs that I've been talking to you about, they're funded in large part by a Massachusetts line item secured by our lead legislative champion, Senator Tarr and Representative Wynn, as well as Newburyport's Representative Shand and many other legislators in the watershed. We also have a new program that just got started, brand new. It's funded through a federal EPA earmark that was secured by Congresswoman Lori Trahan. This program will respond to pollutant hotspots and conduct iterative testing to identify the source of the pollutant. We're still in the methodology approval process and hope to be launching our first investigation in the spring. And our final water quality activity, this is us partnering with UMass Lowell in a research project to test handheld sensors that allow people to test their tap water at home. So that's our water quality work. Another area that MRWC focuses on is regional planning. We're always looking through a watershed-wide lens with a desire to prioritize projects that will have the largest benefit regionally. We undertake complex year-long data gathering and planning projects, like the one shown here for the lower part of the Shawsheen River. This was completed and approved by the Mass Division of Environmental Protection in January of this year. After analysis of the data, priority projects are not just identified, but engineers are hired to draft 30% plans for these projects. Here we're looking at a retrofitted parking lot to better contain stormwater runoff at Andover High School. There are 2 long tree trenches. You can see one in the middle of the parking lot and one between the parking lot and the tennis courts, and a bioretention storage area. And that's more of that design. MRWC also leads a Massachusetts Division of Ecological Restoration Regional Partnership Program. Our partners are the Merrimack Valley Planning Commission, Groundwork Lawrence, Lowell Parks and Conservation Trust, ORS, which protects the Concord, Assabet, and Sudbury rivers, and the Nashua River Watershed Association. Together, we prioritize regional restoration work. MRWC has funneled hundreds of thousands of dollars to these partners to do on-the-ground restoration work. So, that's what we do. Let's take a moment just to be clear about what MRWC doesn't do. So, just to be clear, we have no authority to govern CSO discharges. We have no authority to hold polluters accountable. We don't control or permit dams, and we're not the permitting authority for any other kind of activity on the river. And I throw this out there because you would not believe the number of people who reach out to us assuming that that's our responsibility. We are advocates, not legal enforcers. We use science to highlight pressing issues and try to help point us in the direction of the most efficient and successful solutions. Also, we are keenly aware that we're not addressing all the pressing issues. MRWC primarily relies on government funding for our work, and each contract is very specific regarding what kind of work can be conducted with these funds. People frequently ask why we aren't tackling PFAS or PCBs or mercury or microplastics. The short answer is that we don't have the funding to do so, though please know we'd love to be doing it. So that was the quick MRWC spiel to give you an idea of what we do as an organization. Now I want to sort of give you a quick background on combined sewer overflows, or CSOs. So just to give you a background for those who don't know what a combined sewer overflow is, I'm going to talk about two different types of municipal sewage systems. A combined sewer system, illustrated at the top, and a separated system. So the top two images outline the combined sewer system, which combines sewage and stormwater into one main pipe to take it to the wastewater plant. The top left image shows that during dry weather, the plant has the capacity to handle all the sewage, and there is no overflow. The top right image shows a heavy rain event where the combined rainwater and sewage is too much for the plant to handle, and the untreated sewage and runoff flows into the river. The bottom two images show a separated system, which has separate pipes for the sewage to go to the wastewater plant and separate pipes for the stormwater runoff to go directly into the river. In both dry and wet weather, the sewage never overwhelms the plant, and there is no CSO. Another thing to keep in mind with CSOs is climate change. We're likely to see more CSOs. In 2024, we had an average amount of rainfall, yet the second highest CSO volume within the past 10 years. This is because the same amount of rain fell in fewer but more intense storms, and the more intense the storm, the more likelihood of a CSO. So for example, imagine 6 low-volume rainstorms resulting in no CSOs because there's not enough rain. Now, with climate change, instead of 6 low-volume storms, you have 2 intensive rainstorms that result in 2 CSOs. Same amount of rain, but now you have more CSO discharge in one scenario than the other. So CSOs are bad for many reasons, but also because they release fecal bacteria into the river, and at high levels, it is dangerous to humans. A recent BU study documented an increase in hospital emergency room visits for gastrointestinal diseases in the Merrimack Valley after CSO events. But it's important to remember that CSOs are only one part of the dangerous bacteria problem in the Merrimack. People and the press tend to focus on it because it has a high ick factor. But it probably contributes less to our bacteria problem than the stormwater runoff. Also, the geographic range of significantly measurable bacterial impact, and I'm going to say that again. Also, the geographic range, how far it travels, of significantly measurable bacterial impact of CSOs, so that's how far you're going to get dangerous bacteria readings because of a CSO. That geographic range is limited to relatively small portions of the river. Okay, this is where it's going to start to get fun. We're going to talk about, to illustrate this, we're going to talk about Haverhill. So if you look in the lower left corner of this map, by the way, this map was produced by an engineering firm hired by Haverhill to model the distance of bacterial impact of its CSOs. In the bottom left corner, you're going to see all these orangish yellow dots. Those are CSO outfalls in Haverhill. If you then travel along the river out towards the mouth and you cross right into West Newbury, you're going to see a small red bar. That is, and you'll see next to it, it says 95%. 95% of the CSOs in Haverhill do not go further, the bacterial impact does not go further than that red bar. That's it. If you travel further downstream and you cross over into Merrimack, and then you go into Amesbury, you'll see a larger red bar that has 99%. That's where 99% of Haverhill CSOs, the bacterial impact will travel. Notice we're not coming anywhere near the harbor. So this data was taken with CSO volume data from 2016, for 4 years, 2016 to 2020. During that time, only 2 CSOs traveled further than that 99% red bar crossing. And of those 2, none of them made it as far as even the Salisbury town line. So according to the engineering study, CSOs are not, the bacterial impact of the CSO is not traveling that far. Now, when we started our 2024 testing, we were going to test that hypothesis to see if that stood ground, and you'll hear later about what we learned with that, but they're limited to a relatively small portion of the river. Now I got to get back into my notes. Also, finally, these estimates are conservative for today's activity. The modeling that was used for the CSO volume data stopped at 2020 purposefully because in 2020, additional improvements were made in Haverhill, which dramatically reduced Haverhill's discharge volume per CSO. And so since they had complete data prior to that, they went ahead and released this map, and it really represents a worst-case scenario. What's actually happening today with Haverhill CSOs is probably not going that far. Okay, so this chart shows the data that they used in that 4-year period. The y-axis, that's the distance in miles. The x-axis, that's the volume of discharge, and all those green dots, those are different CSOs that happened in this period. The hash line, that's the 95% bar that we talked about. The solid line, that's the 99% bar. You'll see that there are only 2 green dots that went beyond that. So, the conclusion, Haverhill CSOs are not likely measurably impacting Newburyport with dangerous bacteria levels. And actually, I want to be clear, I want to really talk about Newburyport Harbor. It may be impacting the first part of Newburyport before you get to where the drawbridge is and the harbor sort of opens up. So most people tend to think that no progress is being made on fixing infrastructure to deal with CSOs, when in fact it's a Herculean, multi-year, multi-decade endeavor to address them. And actually, all 5 municipalities, and when we talk about those 5 municipalities on the main stem that dump CSOs, we're talking about Manchester, New Hampshire, Nashua, New Hampshire, Lowell, Massachusetts, Lawrence, Massachusetts, and Haverhill. All 5 municipalities are working under EPA consent decrees and making progress. Different municipalities are tackling the issue in different ways. Some are focused on separating sewer systems, like we talked about earlier. Others are working on creating massive storage systems to store excess runoff to be treated during drier weather when they have the capacity to do so. Others are simply increasing their treatment capacity, while others are doing a varying degree combination of all of those strategies. All cost a great deal of money and take many years to complete. Okay, that's my primer on CSOs. I'm now going to turn you over to Jose, who's going to walk you through our 2024 water quality data itself.
Hello. All right, thank you, Kurt. So, I'm going to talk a little about the data that we collected a little more specifically. Though first, with that, there are some limitations to the data. Firstly, with CSOs. Now, CSOs are hard to plan around. Unlike our baseline sampling program, where I pre-select dates, CSOs quite literally are dependent on the weather. My volunteers are great, and I love them, and they're super committed, but I can't just call them out on a whim after it rains, nor am I available all the time anyways as well. So the data you'll be seeing, all of the data points from the CSO testing, those have a delay of about 14 to 36 hours from when the CSO initially hits to when the sample is actually collected, because of the expanse of the watershed, getting all the equipment ready. Yes. Also with that, in CSOs and for the baseline data that I'm going to show you as well, my volunteers, again, are super great, but I don't want to risk injury on their part. So whenever we have extreme cold or heat, or, my volunteers have lives, so if they just can't sample that day, I miss that data. Across a year, I do collect enough data for it to be significant and readable for the span of a year, but I do notice that whenever I do miss a data point, it does cloud the overall picture a little. As Kurt had mentioned, the CSO monitoring program is actually pretty intense. While it's not a one-person show, I myself am one part of the water quality monitoring program. And we just don't really have the capacity and the resources to test more than one CSO per week. And what happens with that is, for example, if we have a small little storm on Monday, we go out and test Tuesday, Wednesday, Thursday, but then if we get, a lifetime of rain on that Friday, we lose that more significant data point just because we don't really have the capacity to go and test for that. And then finally, a really big picture is actually the flow data for the Merrimack River itself. Starting next year, we're going to try and better use flow data that we can find on the internet. But that's a really big point of monitoring for bacteria generally that I feel like the public sometimes doesn't really take into consideration, or at least not something that's thought about super hard, is just how much volume is coming out from the river itself. And hydrology itself is actually very complicated. We know, obviously, the river starts way up in New Hampshire and exits out at the mouth, but once you try and zoom in, things get complicated, and it's not quite straightforward. Right, so speaking of that, here is our grand map. This is showing our, at the time, 14 sites. We have now added 2 sites in Nashua. As we had noticed, we had a really big gap between Manchester and Lowell for sampling, and Nashua itself does contribute CSOs. We've added that for 2025, but we're looking at 2024 data here. And you see we have blue markers and we have yellow markers. That matters because the Merrimack River is tidal. It goes up to about Haverhill. And our issue there is the specific fecal indicator bacteria that we're looking at. So Kurt had showed you this slide earlier, and give you some more time to look at it. We have 2 different kinds of indicator bacteria that we look for public health purposes in regards to the water. We have E. coli to your left and Enterococcus to the right. Now, Insofar as the downstream and havrel and storm surge might be concerned, we care more about Enterococcus, which is our green guy on the right. You can find both of these in fresh and in salt water, and you'll normally find them together, but Enterococcus survives better in brackish water, like you would get when you get the tidal mixing from the ocean and the river, like you would in the mouth and in havrel. So you'll be seeing mostly data around that guy. The only exception will be for our floating sensor, for logistics reasons, actually. There's a lot of issues with logistics. We can't test Enterococcus with the sensor because those cartridges, there's two different cartridges for the bacteria. Our E. coli cartridges are actually made in Europe, and we can't get them here quick enough before they expire. So we only test E. coli with our floating sensor. Everything else downstream is going to be Enterococcus. All right, that's smaller than anticipated. So, Kurt had gone over this already, but that initial study by the engineering firm CDM Smith, that was actually revolving around where they were going to place signage to warn people along the banks of the Merrimack where the CSO influence actually starts and ends. But we were able to look at that and basically kind of extrapolate, well, okay, if they think we can get this far, the influence stops here, then logically that's probably where the bacteria stop as well. Let's see. So I can give you some more information about their data set generally. So they had 200 individual events that they had collected from havrel when they had built this data set. Many of them were less than 200,000 gallons. For a frame of reference, their largest individual event was 19 million gallons released by havrel, which was more than twice the next largest amount. So that was a very, very significant release. And when they were placing the signage and determining where and how far they want the signage to be geographically, they settled on the 95 and the 99 because they think events like that 19 million gallon release are so few and far between and such an extreme. It's a little safer, I guess more conservative, to select the 95 and 99 points at 6.7 miles and 9.1 on the river. Though this is also something, as I'd mentioned earlier, flow rate is a really big thing that needs to be considered with this stuff. So I do a lot of small life, basically, so bacteria, Enterococcus, things like that. This is much more of a physics problem. It's a numbers problem, where they were taking the data they had gotten and created these maps. Let's see. Also, as well, with the very large releases, reading through that report, I found that only 3 of those events had actually lasted more than 2 days. So 2 days was actually the largest that they had calculated a bacterial plume might survive in the water. I had seen that it could last as little as 6 hours, a significant reading of bacteria in the water. And that's because of the dilution. The middle of the river and any kind of physical water body that isn't a human body isn't great for these bacteria. They don't survive very well. I have done the math myself, and one E. coli, singular bacteria, that replicates to about 8 in about 8 hours, I believe. It turns into 8, and then each of those 8 become 8 more. And that's in perfectly ideal conditions. Out in the wilds of the river, where you get all this turbulence and the different temperature changes, that's not great for them. Right, so I guess with the monitoring program, I wouldn't say we're in our infancy, but we're definitely running into deficiencies in just data and the ability to collect. And that's making it hard for me to like really make draw really firm conclusions. But generally, I'd say I have 3 conclusions that I feel pretty safe about. And the first one is basically rainfall is kind of messing everything up. So for example, this is our chart from 2024, and this shows our 2 data points for our 2 programs. The one to our left is the baseline monitoring program. So as Kurt had mentioned, that is the doctor's visit, regularly scheduled programming monitoring program. And the one to the right is our CSO monitoring program specifically. So you'll see on the left, we have some elevated numbers. And if you see to the right, it might be hard to see. There are 2 bars on the right side. The black one that goes all the way up, that signifies an actual CSO event, not the actual reading. That is the blue next to it. The blue next to that shows you the reading that we had determined. And you'll see during the baseline monitoring program, we had some high numbers. And in the CSO monitoring program as well, as we anticipated, there are some very high numbers. But we're running into the issue with rainfall. Some of these numbers from our left graph were during rain events. Now, we all know CSOs contribute bacteria into the water, but even just general runoff does as well, things that are coming from the land. And we're not really able to distinguish that in either of these 2 programs, because it's the technology exists, but it's just not something that we have the funding to quite do yet. So looking at data points like this, it's a little hard to say if it is actually CSOs or not, especially considering the engineering studies by CDM Smith are suggesting that CSOs may not make it all the way down to the mouth. And this is Salisbury Beach. This is about half a mile from the toothpick. And even during the dry periods, even during our baseline monitoring, we're catching high readings. And we're a little unable to distinguish. Let me show you another one. This is the Route 1 bridge, just near Michael's Harborside. So here, I want to think a little more geographically with you guys. So the Salisbury Beach site was near the toothpick. We're a little further up into the mouth here at Bridge Road. Our left side readings actually look pretty good for the baseline. There's 2 high readings, but generally I'd say it's all right. And then for our CSO one, I'd say it's about the same versus Salisbury Beach. And again, this is all taking into consideration, at least for the CSO side and our right side, there was at least a 14-hour delay between the release of the CSO and the capturing of the bottle. The engineering studies suggest that the bacteria aren't living quite as long as people are thinking, or as we've led to believe, and we're still getting these really high readings. So now I'm going to go to one more. And again, thinking geographically, this is Ferry Park in West Newbury. Haverhill is upstream of all these guys. West Newbury is the closest site to Haverhill that I'm showing you guys right here. And I want to focus more actually on our CSO one on the right. And you'll see, compared to the other 2, we still get like 1 pretty high reading. But, caveat, we have switched to E. coli in this chart, but the 2 safety levels are about the same, even though they're different bacteria. You'll see that the readings of bacteria for Ferry Park, which is the closest site to Haverhill, are much lower. But if you look at our previous 2 sites in Newburyport and in Salisbury Beach, the ones on the right, the numbers are much more numerous and exponential for really? I'm not sure. So, as I mentioned, like this is a lot more that we need to go, a lot more testing on our part. We need to go into a lot more research. We're just getting a lot of mixed signals, basically. So we can't really distinguish between bacteria that's coming from runoff and the bacteria that's coming from the CSOs. And then we can't distinguish between human bacteria, human being red CSOs, things that are strictly from sewage releases, and any other E. coli and enterococcus from any other warm-blooded mammal, like birds and dogs and things. But with all that, there's also this third unrelated, non-runoff, non-rainfall thing that we found actually in Newburyport Harbor with the use of the sensor. So, our sensor has these cartridges that are actually what's collecting the water and running the sample. This whole machine basically is a mobile lab. It's very cool. But back in 2024, it was our first run through it, and I guess we just had too many cartridges at the end of the year. They were all going to expire anyways, so we decided to shoot them off for 2 weeks and see what happens. And you'll see on this chart, all the way to the right is the rainfall for the period between October 21st and November 3rd. It had only rained once on the 30th of October, and that was about 1/4 inch. But you'll see in the results column, there are unsafe readings when it was dry. And that stumbled us for a while. We even went and talked to Fluidion, the people who designed this machine, and we asked them, hey, what's wrong? And they looked through the machine and looked through the data and said, no, this all looks real, it all looks right. So it wasn't raining, as CDM and as our data is suggesting. CSOs aren't really reaching the mouth. So this is some other third problem. There's so much noise going on in the mouth of the Merrimack, as well as all through the other stretches, but right now we're focusing on the mouth, that I'm not quite sure what's happening, and we're looking to try and figure that out ourselves. So hopefully with some more testing and some more resources. I've been asking and coordinating with people in our partnership, those who have done this a little longer than me, and we can try and figure out what's happening in the mouth. And actually, our hotspot monitoring program will probably, this is a very high candidate for it, I'd say. It's safe to say, right, Kurt? So, yeah. So, yeah, it's quite a big puzzle. That's all I have.
So the takeaway for us really is from 2024 data is that we need more data, especially in Newburyport Harbor. It didn't respond the way we thought it would. We had high bacteria readings in dry weather. We also had it in wet weather. Is it a confluence of multiple different factors? We also have a much larger body of water that's also being impacted by the estuary impact from the ocean, and we're not able to test that much. We should really to be trying to answer this question. We need much more robust testing. We need to be testing in dry weather as well as in wet weather and throughout the season. This year, 2025, we were able to sustain the same testing levels that we did in 2024. Look for the report in the spring. We hope to be getting it out much earlier this upcoming year. Unfortunately, given the current funding environment and a stream of funding from the federal government that's no longer there for us, in 2026, we're likely going to have to reduce our testing rather than expanding it to try to answer these questions the way we'd like to. But what can you do? We were specifically asked to respond to this question. So this talk was supposed to be around CSOs. And so if the question is, what can you do about CSOs? And you're living in Newburyport, in Salisbury, there's nothing much you can do about CSOs. If they were to be getting here, they're coming from Haverhill, so there's no direct action step for you to take on your personal property in this community to impact the CSO unless you're doing larger advocacy. If you are so concerned about CSOs, even with the data suggesting that CSOs aren't making it into the harbor, you could advocate with the federal government, with the state government to help fund these municipalities so that the work that they're doing to address their infrastructure can move at a faster pace. But remember, CSOs are only part of the larger dangerous bacteria problem, and there are steps that you can take to reduce the impact of stormwater runoff bacteria. You can install rain barrels and rain swales. You can decrease impervious surfaces on your property. Don't use synthetic fertilizer. Pick up your pet waste. That seems like it's a very small thing, but there was a study done in Seattle with pet waste representing 20% of the bacteria in their rivers. So it actually can have a large impact when it's multiplied. But other ways you can help is you can also get involved with MRWC. You can volunteer with us as a water quality sample collector. And that's actually really important. There are days when we do testing that we lose Jose or Sesha for the whole day because they're having to drive from our office in Lawrence all the way out to get a sample at Salisbury Beach. If from this part of the river we had a robust volunteer crew so that we never had those absences, we'd have more staff time in the office and we could actually support more testing days. We might be able to cover some of the reduction that we may be looking at. Another thing you could do is you could volunteer with us as a culvert assessor. Or if neither of those float your boat, you can volunteer with us as a fundraiser. What you're seeing up there is our upcoming winter plunge. We do it off Plum Island in late January, early February. You raise money from your network of friends and you have a fun time jumping into the water in the freezing cold. Get your picture taken, get in the paper. If you don't have time or the inclination to volunteer, we'd also greatly appreciate your financial support. Regardless, we have a sign-up sheet in the back where you can sign up, either let us know you want to be on our mailing list or volunteer, and we'd be happy to sort of include you in our work. I'm realizing that in this deck there's no contact information, especially for people who may be watching this on TV or YouTube. So our website is www.merrimack.org, and there you can find information for contacting us and joining as a volunteer. And with that, we're going to open it up and take any questions. Just like Kathleen, do you want to come up here and moderate this in any way, or do you want us to handle it? There you are.
No, go ahead. That's fine. But just go ahead and remember to repeat the question.
All right. Also, we only have 2 copies of this here, but these are the reports. They're kind of hefty. The water quality reports. It's available digitally on our website. First, it's going to talk about the different ways that we do our testing, but a big part of this is there's a profile for each of the different 14 sites that we test at. And it'll show you the results in the way that Jose laid out for you for those sites that he showed you, plus it'll give you a narrative on what's sort of going on in that region to give you some context. And it's really pretty interesting how the different sites vary differently. And we focused on the harbor here tonight because of where we are geographically, but it's also one of the 2 largest problematic areas that we found in the river in our testing in 2024. Okay, so questions?
Now that you've seen there's contamination in Newburyport Harbor, have you ruled out the Newburyport wastewater facility as a possible source?
So the question is, now that we've seen contamination in Newburyport Harbor, have we ruled out the wastewater treatment plant for a possible contributor to it? No, we have not. We've not done any work yet to try to identify the source. The hotspot investigation program, because the funding that we currently had was to do regular ongoing testing, not to do specific hotspot investigations, which actually we are now funded. We just received the EPA contract. We're now going through the approval process for our methodology. We expect that to be approved by the spring, and Jose will be launching his first investigation, hopefully this spring, and we'll let you know about it when that happens. But we haven't been able to rule anything out yet. We have lots of different hypotheses as to what could be contributing to it. That might be it, but we haven't been able to do the testing to rule anything out. Yes?
Does any part of your testing include testing of the solids that are in your CSO situation, such as, I hate to be blunt, but there's a lot of other stuff that comes in with a CSO than just the water. There are condoms, there are female applicators, there are teeth picks.
Syringes?
Yes, and syringes. And I've been a volunteer for Clean River Project for 12 years. I've cleaned up homeless camps, and I have to say that maybe the surges in your CSO levels could have something to do with homeless camps dumping into the river. They take their mattresses and they dump them in the river. All of their food and trash, we went around when you're with barrels and needle buckets to try to get them to keep that out of the river. So I just am curious, do you test, when you do your testing, are you testing for what is in the water as much as you are just the water on a chemical level?
So I'm going to let Jose jump in on this as well, but I can start by saying when we're doing our 3-day CSO testing, we're taking a grab sample of the water and testing the water for bacteria. But do you want to add anything to that, Jose? Oh, sorry. The question was, when we're testing for CSOs, are we also testing for other solids that might be in the water as well, or just the water?
In terms of visual, large solids like that, no. So for the actual procedure for sampling for the bacteria, we're using a 100 milliliter grab sample bottles, literally about this big, and our program's strictly focused on the bacteria itself. I definitely don't deny that there might be solids that are like large and floating around in the water. Perhaps it's something we can take on. I try to not give my volunteers too many extra things to do when they sample. This would be like on a data sheet or something. But no, we don't test for anything large like that, though it could be documented.
The only reason that I bring it up is because these plastics, microplastics you were mentioning, you don't have enough funding at this time to get into studying the extent of microplastics. But when all the sewage is coming down the river, or all of the dumping that's going into the river is coming down, if there's no curtailment of it, it will end up getting lodged in the riverbanks. And then it's just like you get a little bit more rain, it's there. Microplastics take thousands of years to break down, but they don't go away. That's why you have PFAS. You have a very strong contaminating situation, and that's why, of course, I'm all for the booms. The Clean River Project tried in, I think, 2016 to go to the state. We went to the State House, and we wanted to get all of the cities and towns to come in as a coalition. Because what goes on up in Manchester ends up down in Newburyport. And, there are different things in the river, but everyone, if we all came together, you could keep a lot of the plastics out, a lot of the illegal dumping not happening. And I encourage everyone in here, if you want to learn more about me and them, to look Clean River Project up and what they're doing on the Spigot River right now, because that directly pollutes the Merrimack.
Thank you.
Thank you.
Other questions? Yes. Yulan.
This is not a technical question at all. I'm just interested in a typical summer month, 2024, with a lot going on. What is a month of testing cost? I mean, are we talking 20,000? Are we talking Just
Yep. So the question, thank you. The question is, how much does a month of testing cost in an average summer month? That's going to be really hard for me to answer, Lon. I'd have to do a whole Excel spreadsheet budget for you. It's also hard because there's the,
If I give you 100,000, how many months would it last?
so,
How about that?
well,
Give it to them, let them try.
well, let's talk about it more over the course of the year, because if you're going to do it, whatever you fund has to be sustainable through the year, because it's got to support the staff people that are going to support it. So that's how, honestly, I was on a very productive call today with Representative Shand and Senator Tarr, talking about this report, and Senator Tarr turned to me and said, what would it cost to do more robust testing in Newburyport? And I said, okay, Senator, give me a week and I'll come back to you with an Excel spreadsheet. But that's how I think about it, is how to support it through the season. And
That's why I said summer, because you don't have a massive program going in the winter.
the actual supplies to do it's real. I can't answer your question, Lon. It's impossible. The supplies are a cost, but they're not that outrageous of the cost. It's more the staff to coordinate the volunteers and run the testing. And when we test, there's the day of actual testing, and there's the day before for the planning and the prep of all the logistics. There's the day of with collecting the samples and starting the initiation of the testing. There's the day after that's when we get the results out of the incubator and the reading of it and the posting of the results. And that's just the actual test. It's not compiling it into this report and doing all the extra work that goes with it around it. And so there's staff time that's needed, even though we're heavily reliant on volunteers. And that's why there's a request for volunteers, because the more we don't lose Jose or Sesha to go out and do that, it reduces the cost of the program. Typically, for what we currently are doing, the baseline and the CSO testing, let's say that costs around $250,000 for the full calendar year.
Okay, that's, I can divide that 12. No, that's good.
That doesn't count the sensor, which is outrageously expensive in and of itself.
I'm just wondering if you could piecemeal a program like that together, instead of having the Fed stand you a check for $220,000.
We actually do piecemeal it together, and that's why we have to kind of think about it layering.
Yeah.
And unfortunately, when we lost that federal stream, that was part of our foundational work of our baseline program, which we couldn't add anything else on top until we filled in that gap, because everything sort of relied on that foundation to be able to do the additional components. So it's a complicated budget picture. Next question.
Is the testing done in the field, or do you just get the samples back to your office or lab and do all the work there?
So the question is, do we do the testing in the analysis and the testing in the field, or do we bring it back to our lab? And I'm going to hand that over.
It depends. For the CSO monitoring program, that one is done in the lab, where either I or a volunteer would grab the bottle of water for me. I would take it, and then I would run it through and add some reagents to do chemical stuff to it, and then run it into our incubator and through our system. It's called IDEX, that basically spits us back a number as to what it thinks the bacteria concentration is. I only say it depends because for our main stem sampling program and for our watershed-based plan programs, when we were doing those, we did have testing equipment that you would actually put in the water, and you would give you a reading instantaneously, and you would write that down. But I would still need it eventually, so it's a bit of both.
I'm asking because I'm wondering if you were able to get regular samples sent to you, if that would be helpful.
I'll take it if you want.
You want?
Here you go.
I've definitely been asked that a lot, and more data is always good, but the issue is you need to go along with the data. Our 13 sites, our 15 sites now, was 13, before was 8. Some of these sites you've been testing at for over 8 years, and it's only around then that a piece of data, or at least a data set, is considered good. You need to go like really long sampling just one place for a while. And it also depends on what the project is exactly looking for as well. So.
And I would say that any given site on its own, we're probably not going, it would have to be part of a coordinated effort and done in a timely way. When we're collecting samples, they have to get into us within a specific window,
6 hours.
6-hour window. And then they're all run as a batch. So it would have to, if you're interested in volunteering as a water quality collection person, you'd be really more incorporated into a program and working in whatever batch is being run at that time, as opposed to many different people sending us in samples, even if that's on a regular basis. Next question. Yes.
You
Yes.
mentioned that you lost your stream of federal funding. I was wondering if you could tell us, like, the main agencies or divisions of the federal government that have cut off the funding for
So-
this effort.
So the federal funding that I was mentioning that we lost was not due to a shutdown or the new administration. That was a grant from the EPA that was funneled through Mass Bay's estuary program. It funded some of our watershed-based plans that we were doing, as well as our main stem foundational work. And that grant ended as of the end of June. So there were other sources that we were hoping to be going towards, but the funding environment has contracted. And so with that, we're looking at having to pull back and conform and live within our means, and our testing will be somewhat reduced. And we're still waiting, excuse me, to see what landscape we're looking at in February to really lay out what our plan is for the upcoming calendar year season of testing. By the way, for those, the question was what federal funding was cut. I feel like we're playing Jeopardy. There's the answer. Now the question was. Next question, yes.
I'm confused.
Welcome.
So correct me if I'm wrong, it seems like looking at the data from the Haverhill CSOs, one in Newburyport Harbor might not be terribly concerned by CSOs from that particular plant. I realize there are other plants that are also pumping volumes, but let's say it doesn't reach the harbor, it sounds like. And yet, you have a monitoring device near the harbor master's office where we were incubating oysters this summer, by the way. They grew very well. And that shows concerns about bacteria, about dangerous levels of bacteria. Give us some ideas of what you're thinking. I mean, isn't that just totally, is it coming in with the tide? I guess that's the question.
So this is where I hate to throw out what my ideas are. Because you're going to latch onto the one that you think sounds right, and when we finally are able to test for it, that might be the first thing that's ruled out. It could, here I'm going to go, I'm going to maybe throw it out there. Let's go for one. It could be illicit connections where people have their sewer line hooked up to the storm water system. Could be that, and it's coming through the outfalls. Maybe there is some kind of a breach at the wastewater treatment plant that's not currently detected. Could be coming from boats discharging. Who knows? Until we can do the testing and get in there and identify it, we're just guessing. I mean, we honestly, when we started in 2024 with this part of the project, part of it was to go in to say that engineering study, is it really true? We kind of weren't quite sure we were believing it. But our data kept supporting it. We weren't seeing readings in Ferry Park or at Deer Island anywhere near these inflamed readings that we were seeing in the harbor. It really seemed as if the CSOs just weren't making it that far down, and something else completely different was happening in the harbor. And then that was further substantiated by all the sometimes exponential readings we were getting in dry weather. So we're confounded. We want to do more robust testing. We currently don't have the funding to do it, but we're looking to see what we can get. And by the way, the investigative hotspot investigation program, that's not going to be able to really tackle the Newburyport issue. It might give us a first level scan, but the way that that funding is written, it's imagining a hotspot is a single isolated issue that might be coming from someone dumping in a storm drain. So it's each hotspot investigation is contained to 6 iterative tests, approximately 30 tests, and then a report is written. And we get 50 of those with this funding. Newburyport, if we tried to solve the Newburyport Harbor, that would eat up this entire funding, and that's not what this funding source is pointed towards. We're probably going to be able to utilize it at each of our current testing sites, calling each one a hotspot and writing a report after 6 iterations of testing, and that's going to teach us something, but it's probably not going to identify the source, especially because, at least here's my gut, here's something I will say, I don't think it's one thing. I think it's a confounding, it's so cloudy to us, and we're not able to make complete sense of it because there's more than one source of something going wrong here. And trying to tease it apart and identify that is going to be a task.
You have a question in the back.
Yes.
I have a question about your data sets. This 2024 data is about bacterial information. Are your historical data sets also about bacteria, or are they measuring other things?
Okay, so the first question is, are our legacy data sets dealing with more than just bacterial information? And before I hand it over to Jose, the report, what we were focusing on right now was the bacteria in Newburyport Harbor. The other parameters that we test for in the baseline testing, those parameters are in the report as well. We just didn't cover it because it wasn't remarkable. It pointed towards a healthy river, and this seemed like the topic to cover. So if you go on our website, you look at the report, you'll see a whole section on that, and you'll see the results for that as well. But with that, I'm going to now hand it over to Jose.
Yeah, so MRWC was founded in '76, I believe, and I've been going through a lot of our old data sets and what we still have in physical paper. A lot of it's been lost. We were still doing bacteria back when, but we were also doing a whole lot of other things back then. But we had just contracted, and now we're back as we are today. And we're honestly kind of just rebuilding capacity because, you know, we're called the Merrimack River Watershed Council. We used to expend testing all the way up to the White Mountains, like Franconia Notch and things like that, and doing all kinds of monitoring, not just bacteria. We're doing, like, you know, macroinvertebrates, a lot of the baseline parameters like temperature and things like that. Though currently we're focusing more on bacteria and just the baseline parameters, as I call them, just the basic physical chemical, like pH, temperature, salinity, just because those are fundamental to getting an understanding of any body of water. Those are generally the first things you would want to try to get, those physical chemical parameters, and then bacteria, and then you would move on to more other important things like nutrients, macroinvertebrates, because each of those different types of bacteria have a different like time frame of like usefulness. Yeah.
Yes.
I have 2 questions. One, you said the automated sensor in the harbor is super expensive. What's the range you're talking about there, and are you able to run it as much as you'd like? And then the second question is, has the city been involved in the discussions about the harbor data that you're seeing, and what was their response?
Okay, so let's start with the first question. And that's, I said that it was super expensive to run the sensor, so can I retract that? So that question was, how expensive is the sensor? The sensor is expensive, and one of the parts of us exploring the sensor was to explore the cost. And we have found, although the sensor is a really exciting thing to talk about because it's new technology, they used it in Paris for the Olympics, and we're learning about cutting edge, whatever. It is much more cost effective for us to do the volunteer-based sampling. You think that, oh, gee, you have this remote sensor, it just does it, you don't even have to go there. Well, you have to go there at least once a week to change the cartridges. You've got to go there as well to change the battery, because it goes down.
Yeah, clean out the barnacles.
You've got it is its own set of human hours, plus the cartridges are very expensive. Especially if you were to use Enterococcus cartridges, which come from across the ocean and have a shorter lifespan and have to be refrigerated while they're being and you have to send it every year in to be calibrated, which you're shipping this huge heavy thing all the way down to Texas, having it be calibrated at a cost and then ship it. So there's a considerable expense. Before Hal has a chance to ask me, Lon, excuse me, I'm going to guess that it's about per sensor just for maintenance and use, $15,000 to $20,000 a season. Plus, if you're purchasing one, they're around $30,000. And we are lucky to have ours loaned to us, but we're still having to come up with the cost to do the yearly maintenance. The second question was, is the city of Newburyport involved in the sensor? They actually are allowing us to use the pier. I don't think I got that question.
Aware of the data that you've found in...
Is the city of Newburyport aware of the data? We've shared it with the Public Health Department. We've had conversations with them. They're concerned, their eyes are wide open, and they're engaged in discussions with us for what else we might be able to learn.
How exactly does the bacteria have an effect on human health? Does it have to be ingested in order to cause harm, or does it have an effect on marine life, or how is it transmitted?
Are you?
Yep.
Repeat the question.
Yep. So the question was, how exactly do the bacteria harm human life? So basically, it can be harmful in two ways, as you suggested. The first one, being ingested. So these bacteria, they actually are found in the human body and warm-blooded animals generally, but they become an issue because they get excreted. And it's not even the E. coli and the Enterococcus themselves that are harmful, potentially. There are other strains of E. coli and other bacteria generally that are harmful to humans and can cause gastrointestinal issues, vomiting, diarrhea, shingles. And then also if you have like open wounds, they might get in there and cause a rash as well. So.
Yeah, one thing I'd also add to that is BU, when they did their recent study and they actually looked at the Merrimack River and were analyzing emergency room hospital visits after CSOs, and there was the spike in emergency room visits. That's basically what they could say, but we don't know how it led to that. There's no understanding. Is that what did the moisture rise in the air? And who knows how that translated into those gastrointestinal emergency room visits. So there's an element that we know. There's also elements that we don't know yet. Other questions? Yes?
When you were testing in the harbor and you're getting these high readings, do you account for whether the tidal flow was coming in or going out when the elevated levels were detected?
So the question is, when we were getting high readings in the harbor, were we accounting for whether the tide was coming in or out? Do you want it, or I can?
Yeah, I can do it.
Yep.
Yeah, so as Kurt and I both alluded to with the Fluidion, and as I mentioned earlier, there's two different kinds of bacteria. The Fluidion has two cartridges, one for each type of bacteria. The Enterococcus one, which I'd say would be preferred generally for the downstream because it's brackish. It's too expensive and logistically it just doesn't work. So we have to use the E. coli ones, but the E. coli as a bacteria, they're not as hardy as Enterococcus just has to do with like the shape of the axle bacteria itself, that it just can't handle salt levels. And because of that, it's not useful to us to test E. coli in salt water. So we explicitly have to time the machine to catch low tide on one of the 2 low tides in a day. So that's what we do for that.
So that's for the, so that's for the sensor readings. The baseline readings, Jose has to schedule all of our baseline testing a year in advance based on the tides, and they go out and test in a low tide window. So for our baseline, that's all consistent. When we do our CSO testing, we don't have that luxury. We're going out at the same morning window for 3 days after a CSO. So that doesn't get to account for the tidal influence. Currently, with our resources, that's the extent of what we can do.
Could other, currents from the ocean coming up from Boston, pollution coming into the Atlantic, and then somehow or other, is there a current that could carry it up into Newburyport?
So the question is, could the high readings be coming in from a current coming in from the ocean? And again, I'm not going to rule anything out yet, but from what I'm seeing from engineering studies and what's available to be coming in within the distances that might be able to impact it, I'd say that's unlikely. But until we test, we wouldn't be able to rule that out, and we'd be looking for what's coming upriver with the tide coming in, as well as what's going downriver.
Do you see a spike in emergency room visits after a CSO? There's a lot of recreational activity upriver, like rowing clubs and sailing clubs and so forth. Did you see after there's a CSO? Is there a spike in emergency room?
So I'm just because I don't want to take responsibility for it because it's not my study and I can't speak for it. But BU did. Oh, sorry. The question is, do we see a spike in CSO-related hospital emergency room visits after CSO, gastrointestinal issues? And in the BU study where they evaluated emergency room data and timed it out with CSOs, they did notice a spike in gastrointestinal emergency room visits after CSOs in the Merrimack River Valley. Did that answer your?
Yes.
Yeah. Okay. Yes.
Are you recommending that people don't swim then in the basin at all, just based on this data?
So I would say that it's up for everyone to make their own decision for what. Oh, sorry. Am I saying that I would be recommending that it's not safe to swim in the Newburyport Harbor Basin area? And I would say it's up for everyone to make their own decision as to what is safe and not safe. We specifically try not to tell people that. We say, here's what the EPA is telling you is a dangerous or safe level, and this is what we're seeing happening. There's also, our data is continuing to suggest that it is extremely challenging, if not impossible, to predict what you're going to have for bacterial impact after any given CSO. Each one is different. The volume, the intensity of the rain, the length of the discharge, the volume, the density of, was it a big dump in a short period or was it a long discharge over a long period of time? The flow rate, the other stormwater runoff. Was there a CSO 2 days ago? All of that, it's impossible to predict what's going to happen. Plus, we see things that are not CSO or rain event as well. All we can do is provide you with the data and let you make your best decision, and we're not giving a recommendation at this time. My eyesight's bad, so I don't see the people in the middle of the back, so why don't you go ahead? I've probably been ignoring you. Yes, you.
I wanted to know if you seasonally tested, one of your things was to say, could the boats contribute? Did you see a higher spike in the boating season? In other words, from April until October? I mean, did you take that into account? I know you're guessing at that.
Yeah. So the question is, are we seeing a higher spike in the boating season? And I guess what you'd be saying specifically in the harbor, not throughout the river. I'll let Jose answer. I have my own thoughts as well.
That's kind of hard, if I'm honest, because there is a seasonality with the bacteria. I know you're talking about the the mouth generally, but even across the whole river that we've been testing. As the water gets colder, doesn't mean it's going to kill the bacteria, but we definitely see lower levels throughout the winter, and then they pick up in the summer. And so far as relation with the boats, that was a theory. It's a theory that we have among many others about what's happening in the mouth. It just takes a lot more testing, and that's really an enforcement issue. And MRWC, we have no enforcement power, not legislatively.
Yeah, and what I would just add to that, it sort of underscores what Jose is saying, is that the bulk of our testing occurs during, what you would say is the boating season. So it's hard to compare unless we had more robust testing as well in the off-season to be able to do that comparison.
But it's interesting that month data that you had was October and November, right? So that was post-boating season.
So this came up in a conversation earlier today, that 2-week period was the last 2 weeks of October, basically, when a lot of boats would theoretically have been starting to be pulled out of the water, but there are still boats there. So it's unclear. Again, until we get more data, we're pulling at straws. Yes?
I'm wondering if there is data or research across estuarine areas up and down the coast where you might look at similar types of areas and whether you might find that there's something about an area near the mouth of a constricted river that has something where that happens. I'm thinking about places like Barnegat Bay or down near Savannah and those kinds of areas. There are research estuaries, and I wonder whether they might have some data, not exactly the kind of data you're collecting, but that might point you toward a reason why the harbor and the mouth might have that kind of high levels of bacteria.
So the question is, have we looked into or thought about possibly seeing if other somewhat similar estuary situations might be experiencing a similar type of bacterial experience that might help, and have maybe even started to understand why, and if that might help us in understanding what's happening in the harbor. And I haven't done that as of yet. I'm going to look to Jose to see if he knows of that, but I think that's a perfectly good idea to be studying. The one thing I'd put out there as well is Newburyport Harbor is a very unique and complex hydrological phenomenon, and but it definitely bears, in my mind, looking into.
Yeah, that's not something I have thought to do. I will definitely consider that, but not to bash on Newburyport Harbor, but really every body of water is a unique hydrological puzzle. We can draw comparisons, we can try, though I'm not sure myself, personally, how much comparison drawing we can do between really any other mouth of a river with the Merrimack. But it's definitely something I'll look into. So.
Do we have time for another? Or?
Just one quick question, then we'll go ahead.
So, yes. The one I'm pointing to? Right there, yes. You.
I couldn't help but hear that you really desperately need volunteers. There's probably a lot of people in this room tonight that would be willing to do that. And I would just ask, like in town here, or even along the river, where you have collection points, and there's maybe private property involved, have you thought of just even, or public property, have you thought of, like in town here, the Acres program, where they have the signs along the rail trail? And I don't know if there's QR codes and you can put right there, looking for volunteers to collect. Not that you can also go to the high school and all these places, because there's so many young scientists around here. Maybe they don't have driver's license yet. I mean, most people on rainy days are sitting around waiting for the weather to get better. And they would probably be, a day or two afterwards being, willing to do things. But I know that, I follow you guys on all the social media things. I don't know if that gets to everyone, because it gets pretty busy, but just some ideas.
No, all ideas are welcome. Any ways that we can recruit volunteers, we'll consider and take on. The problem, one of the problems with CSOs is it's that rainy day. That's not the day you need people. You need people the next day when the sun's come out and people want to go out and play that day. And they think, oh, it's sunny, so the river must be okay. And that's actually the day when.
We're not supposed to go in for 48 hours, so those of us who use the river and the ocean, we wait 2 days. Now we know we don't have to. It's going to be dirty anytime.
Thank you, everybody, for joining this evening, and thank you for your thoughtful questions. We appreciate that. There's some more snacks in the back. Please help yourself, and excuse me. Yeah. Thank you again.